how they are stained with these differential staining techniques, and this staining technique is the most frequent one that is used for identifying the chromosomes. And so this staining technique is called G-band staining. And so there are 23 pairs, 2 X chromosomes. Obviously, you can tell this from female. But what is important is that I'll touch upon how this staining is done. But typically, it is done after treating the chromosomes with a protease, and in most instances with trypsin, and then staining with Giemsa. And what it shows you is that there are dark bands and light bands along the length of the chromosomes. And in fact, based on the organization of the longitudinal differentiation in the staining pattern that you see in each chromosome, each chromosome can be identified as a specific chromosome. For example, this is the staining pattern of chromosome 1, which is very different from chromosome pattern and staining pattern of chromosome 6. All right. So, so this is how you identify the chromosomes. But so that's at one level very interesting, right? I mean, for example, we all know that Down syndrome patients have trisomy 21, right? How do you identify? When you see a chromosome that is stained like this, and you have 3 copies of them, you know that it's a trisomy 21. Similarly, there are a number of chromosomal translocations identified in different cancers. Okay, and most commonly known in hematopoietic malignancies, leukemias, lymphomas, which involve chromosomal translocations. And again, those translocations can be identified by staining the chromosomes by this kind of technique. And again, a very commonly, not very commonly, but very frequently detected chromosomal translocation seen in one kind of hematopoietic malignancy called chronic myelogenous leukemia involves a translocation between chromosome 9 and 22. That's the classical case of chromosomal translocation that was first identified, and which is also called Philadelphia chromosome, and it's seen in almost 90+% chronic myelogenous leukemia patients, and that involves a reciprocal translocation between chromosome 22 and chromosome 9. And subsequently, we have identified that this leads to juxtapositioning of 2 different genes from 2 different chromosomes, and as a result of which a gene called BCR is placed upstream of a gene that encodes for a kinase called Abel kinase, as a result of which this kinase activity is significantly elevated in these cells. But anyway, so this has been the starting point of identifying chromosomal translocations, by staining these chromosomes with these differential staining techniques. But also, it tells you that along the length of the chromosomes, the chromatin is organized differently, right? It has to be, and that's why you see this kind of staining pattern. So these are the different staining patterns, like I was telling you, that are commonly used. G-banding is the most commonly used staining technique for identifying chromosomes. What we have recognized since then, that, so like I showed you in the previous slide, there are dark bands interspersed with light bands, right? Or pale bands. And we now know, based on the DNA sequence analysis, these dark bands are typically AT rich, and the pale bands are GC rich. And the R banding is, of course, the reverse of it. And again, there is a reason why these banding techniques are referred to by these kind of names. G-band stands for Giemsa banding. That's the stain that is used for staining these. R banding, it has typically done after heat denaturation. Stands for reverse banding. And Q banding is named like that because it was first done with a fluorochrome called phenochrine mustard. So from that phenochrine mustard, the initial Q was used for referring to this kind of staining. Similarly, C banding is called C banding because this typically stains the centromeric regions of the chromosomes, and that's why the centromeric heterochromatin of the chromosome, and that's why they are called C banding. And so now, this is is where we start getting into the complexities of the chromatin and how it is organized into the mitotic chromosome. So this is a cartoon diagram showing, how DNA duplex is first organized into these coil fibers along the length of the chromosomes, with each individual chromatin unit organized into nucleosomes. And how many of you know about nucleosomes? You know, right? All of you know, I suppose. Do you? Huh? Do you? Yeah. So do you know how many proteins and how much DNA is involved in a nucleosome?
8.
Yes.
Which one? I forgot details.
Right. Anyway, so you know that. So yeah, so we'll show you what those histones are. But we'll talk about it. But anyway, so this DNA duplex is wrapped around a histone octamer actually, and that octamer is made up of histone H2A, H2B, H3, and H4. H1 is actually called a linker histone, which is in between these nucleosomal units. But this, at this level, it's getting coiled into what's called, it's actually of 10 nanometer width, okay? And then it is further wrapped around into 30 nanometer chromatin fibers, and then a scaffold comes into play around which the chromatin is organized in the form of loops, and with further compaction, the 600 nanometer fibers are generated, and that ultimately is further condensed during mitosis into these kind of chromosomal structures. All right. So this is again this staining pattern you do not see with simple staining of the chromosomes, but you have to do so this is actually showing the G banding pattern. Now, again, this G banding can be done at different resolutions. In other words, depending on how condensed the chromosomes are, the number of bands vary. Okay? So this is typically from I believe a 400 band preparation. When the entire chromosomal content of a human cell can be distinguished into 400 band resolution level. Okay? So, and then because of this banding pattern, we are able to now identify each different segment of the chromosome into discrete units, all right? So, and typically each of these units are numbered accordingly from centromere towards the two ends, okay? So you also see this P here and the Q here. You know what this stands for?
The arms?
Yes. So the P arm and the Q arm. And you know why it's called P arm?
Short.
The short one, yeah, the short arm, which is for the petite, right? And because it was called P arm, the other arm was called Q arm, all right? So, so typically, if we want to say where the chromosome may be broken or where a chromosome may be translocated, we can refer to a discrete unit, based on the staining pattern, and say this is where the chromosome has been broken or was translocated. For example, this segment here will be called P211, okay? And similarly, on this side, it will be called Q12. This will be Q211, and this will be P12. So this is a standard nomenclature that has been established, by investigators from all over the world, which is commonly followed by everybody. So that, if I refer to a chromosomal translocation that I see in my lab, somebody else will be able to recognize the same thing based on the same standard nomenclature with which the chromosome segments are named. All right? And this is how the DNA duplex is actually, condensed and organized into a mitotic metaphase structure. And this, of course, in the beads on a string image, from many years ago when it was first identified, where you can, under the electron microscope, you can actually see these coiled chromatin fibers, and also the individual nucleosomal units threaded along the length of the chromosome or the chromatid. So these are each nucleosomal units, with the linker chromatin in between. That's the same structure that we were looking at earlier. So this is how this is organized. The 30 nanometer fiber, and each of these units is a nucleosomal unit, all right? And so like I was telling you, so the histone H1 is the linker histone, which is present in between the adjacent nucleosomes, and each of these nucleosomes have a width of 10 nanometers, and it is actually DNA wrapped around an histone octamer. And what are these histones that are involved in this octamer? Like I told you, this is H2A, H2E, H3, and H4, and these are all histone molecules. And again, depending on the conserved motifs in these histones, you can identify different domains. So typically, all of these histones have a certain amino acid motif that is called the histone fold, and they all have this projecting N-terminal tail, which is called the histone tail, right? And this is how they are organized. So this is the amino terminal histone tail of H3. This is the amino terminal histone tail of H4, and similarly of H2A and H2E. So these form dimers and then tetramers, and then these two sets of tetramers come together to form the histone octamer. All right, and each of them, the histone tail actually projects outside of this histone octamer structure. So again, in another view, this is how it looks like. All right, and so this is the red is the DNA wrapped around it, and these are the histone octamers from which the amino terminal tail projects outside. So again, this just shows the spatial organization, at different levels. Okay, so that's the fundamental chromatin unit, right? And then when you look at the condensed chromatin, then typically there are 3 regions on each eukaryotic chromosome that is responsible for the replication and segregation of this chromatids or chromosomes. And so you have the replication origins, and again, I'm sure most of you know that the replication origins is where the replication starts in a bidirectional manner, which forms a replication fork. And then of course the centromere is a region that we will be talking about in a little more detail today. And then the ends of the chromosomes have a protective covering, sort of, or cap, which is called the telomere. Okay? Because you have to recognize, again, how can the chromosomes be protected from getting degraded? And so these are nature's way of actually making sure that the individual chromosomes do remain as individual units. Okay? So this was actually much before, many years before, decades before the structure was unraveled. The concept was already there, that there must be a protective cap at the end of the chromosomes for the chromosomal DNA to be protected from the degrading enzymes. And subsequently, of course, we have recognized how this protective cap is formed. Okay, and we'll go into a little bit detail of that today also. And of course, but before that, we'll be talking about the centromeres. Any questions? And I'm just curious to know, is it all making sense of what we are discussing? No? You don't have to agree with me. If you don't, you say I don't. But are we learning anything? No? That's good. And why do you think that we are not learning anything? Because you knew?
Yeah, I already know.
So, that's good. So what more do you know about the centromeres? I mean, that you know that there is a centromeric region, right? Do you know more about something more about the centromeres?
It's highly condensed. It's where the kinetochore forms.
Yes.
There's not really any genes there.
Yes.
It's important for them to divide during mitosis.
Right. And do you know anything about? Again, I mean, don't get me wrong. I'm not trying to test you. All I'm trying to find out is, you know, what's the level of discussion that we should be having in this class. I mean, you know, I mean, that's just for that intent. But I mean, are you aware of the centromeric chromatin organization?
Just that it's highly condensed.
Do you think the chromatin in the centromeric region is the same as the rest of the chromatin?
Well, it's heterochromatin. Heterochromatin. So
Okay,
it's not
but in
really-
terms of the proteins,
No, it's not the same.
huh?
I don't think it's the same.
But do you know what it is? Okay, that is something you were maybe there. Yes.
I was wondering if we can get some reading book chapter, if you can give us that ahead of the class that we can read and come prepared, or later we can go and study it.
I think that's a good suggestion. I mean, we have done it in the past sometimes, you know, where I would bring some review articles. Unfortunately, not everything may be there in a book chapter, but we can certainly try to give you some review articles, and I will try to get some of those for you. All right. Anybody else? I like it, you know, sort of, and feel free to express what you think. So, since we are talking of what we have learned and what we have not learned, did you learn anything in the last class? Okay, that's good. So, in other words, what did we talk about? So, you already knew about cyclin CDKs, which CDKs are involved. You already knew about cyclin dependent kinase inhibitors. You already knew about RBE2F function. That's fine. But did you already know about which phosphorylations are important?
A little. Taken an epigenetics class. About phosphorylation, a little.
So, who was the epigenetics class?
It was in my undergrad.
Okay.
I took a lot of genetics courses.
Okay. So, but let's go beyond it. So, I would like to know from the rest of you also, you know, how many of you felt that you learned something in the last class?
I had a problem with the last half of the chapter, last class's chapter. The first part was good. I knew a little, so I was okay with that. But later, I went home, I looked into the book also, I didn't find it.
And which part are we talking about?
That same CDK2 and
The
DNA replication pathways, the pathways and all that. Because I'm the person who is like, I need to read through all this to understand after class. But I didn't find it there in the book.
yeah, so that's the problem, you know. I mean, we don't. I mean, again, we try to strike a balance between the fundamentals and, you know, also give you a flavor of what's going on at this time. And you know, typically, not all textbooks are always, you know, written like that. But we can certainly provide you with some review articles, you know, that cover those topics. And I'll try to get them next week. All right. So, and the rest of you, I mean, I want to sincerely know, you know, who were there in the last class. What was the problem that you felt after sitting through the class, and where do you think we need to improve? And be bold and frank. I mean, there is nothing to be afraid of. I mean, again, the purpose is for, I mean, the whole entire exercise is for you to learn. If you are not learning, then it makes no sense. But let me also tell you that we expect at this level the students to go back and try to read through the literature themselves also a little bit, And it's so easy these days. I mean, all you do is just type your search words in PubMed, and you start getting stuff out. So, but nonetheless, this has to be of some benefit to you. Otherwise, it defeats the purpose. So, feel free to express that. And again, the problem sometimes we have it. You may be coming with significant amount of background, but not everybody does. But we try to build upon it as we go along, and there will be certainly things, on specific lecture topics that would be significantly new. Okay? And so that's what you may expect. Okay? But nonetheless, if you have any questions, suggestions, feel free to express them. All right? Okay. So this is how the centromeric region looks like under electron microscope. So since you mentioned about kinetochore, do you know what kinetochores are made up of?
The big complex of proteins where the microtubules, granules, attach.
Right.
That's what it is. And that's
Okay. That's good. So anyway, so that's precisely what we are saying here. That so this is the region where the microtubules actually attach, and this is the region which is called the kinetochore region, which is assembled next to the centromeric region when the cells are ready to divide, when the chromosomes are ready to segregate. And if you look at it carefully, it looks like a trilamellar structure. Okay, there are 3 units here, and of course, these are the microtubules coming from this end of the pole. Okay, so this chromatid will be pulled to this part, and so these are the microtubules that are embedded in the kinetochore region next to this centromere. And like she mentioned, this kinetochore is actually a very complex structure, and we will go into a little more details of what they are all about. So if you color code them, like I said, this is a trilamellar structure. We call this area as the outer kinetochore. So this is the reverse of this. Okay, so this is the electron micrograph, and this is the color coded image of the same structure. All right. So this side is the outer kinetochore, and this side is the inner kinetochore, and of course then there is the inner centromeric region. So outer kinetochore is the region where the microtubule binding and microtubule motor proteins are actually localized. This is also the region which is involved in the signal transduction that tells the chromatids that they are ready to segregate. The inner kinetochore has the centromere replication protein, the chromatin interface, and this is the region which is responsible for the kinetochore formation. And of course, the inner centromere has the protein that is involved in the chromatin cohesion, and also a set of proteins that are called passenger proteins. Okay? And the passenger proteins are called like that because they piggyback on this region of the centromere, up to a certain stage of cell division, and then they localize to a different region. So these are the set of proteins that have been identified localized in the different regions of the kinetochore and the centromeric region, centromeric structures. So the outer kinetochore has these proteins that are involved in microtubule attachment, regulation of microtubule dynamics, and the checkpoint signal. And again, this is a long list of proteins, but we will be touching upon just a few of them that are critical players in microtubule regulation of microtubule dynamics. So when I say microtubule dynamics, you have to recognize that the microtubules first originate from the spindle poles, okay? They elongate. So the end which is elongating and coming towards the chromatid is called the positive end of the microtubule, okay? That comes and gets embedded in the kinetochore, all right? And similarly, when the cell is ready to divide, these chromatids start shortening, or the microtubules, the spindle starts elongating. That pulls these chromatids apart, all right? So this is a dynamic process, and this dynamic process is actually regulated by these sets of proteins, okay? So you can tell that this is a highly complex process. So these are the inner plate proteins. There is an interzone protein that is involved in tension sensing and checkpoint signaling. Because what do I mean by tension sensing? How do the cells, how do the chromatids know that they are ready to move? One of the things that we recognized many years ago is that somehow these regions are able to sense a tension from the two ends, okay? Because when the microtubules attach to the two ends, then obviously there will be a tension generated, right? If it is all attached only at the one end, the other end is not attached, there won't be any tension, right? So, in fact, there were some very interesting experiments done much before these proteins were identified, where somebody tried to prick this region of the centromere with a needle, okay, to generate an artificial tension, and then immediately the chromatids separated. So there was a physical sensing of the tension involved for the chromatids to separate. And later on it was found out that that protein was called 3F32 antigen, which gets phosphorylated following tension, and that helps for the chromatids to separate. And then of course there are these centromeric heterochromatin associated proteins, and many of these proteins are still being investigated for their functions. And again, we will touch upon some of them. Again, as you can tell, this is a very active area investigation, and highly dynamic and complex process. And it's not possible to go into the extreme details of these processes. But again, I expect you to understand the fundamentals of it, and we'll go into a little bit of that. Let me tell you that we have a paper just accepted in the journal Cancer Cell, where we have shown that this protein, let's see where it is. It's not mentioned here, but anyway, one of the aurora kinase family proteins is involved in inactivating the checkpoint for the chromosomes to move to the two poles. Okay? Yes.
So, okay, when we have in the mitotic cycle, when the chromatids don't split, we have that issue. Is it because the tension receptors don't get activated, or is it because the microtubules are not strong enough to pull them apart?
Both are true. So the tension receptors won't be activated till the microtubules have attached properly.
Okay.
Okay. So one is consequential to the other.
Right.
But obviously there won't be any tension till the microtubules have attached from the both ends. And again, there are different terms, and I don't want to confuse you with too many terms, but the fact of the matter is, if you look at a live cell undergoing mitosis, you do see that it's not an automatic process where every centromeric kinetochore attaches to the microtubules coming from two poles, okay? Sometimes they misattach. To begin with, they attach at one end, okay? And then the same end may be attaching from the microtubules coming from the other end. And so that is a misattachment. And so long as that misattachment persists, there is this, what is called as weight anaphase signal, these are again operational terms given to the phenomena that we observe, which does not allow the cell to go from metaphase to anaphase. Only when they attach the kinetochore, the sister kinetochores attach microtubules coming from two separate poles, two opposite poles, and this attachment is called amphitelic attachment. Okay? Only when that amphitelic attachment happens that the chromatids separate. And how does that happen? And how does that sensing happen? We'll mention a little bit of that. But before that, let me show you the basic structure of the centromeric chromatin. So the centromeric chromatin, we are also showing Saccharomyces formica and Drosophila melanogaster. And again, this is also a fascinating area of investigation. The fact of the matter is, whether it is Saccharomyces or yeast or Drosophila, the fruit fly, or humans, they all have centromeres in their chromosomes, right? But when you look at the DNA sequence, the DNA sequence is not conserved, not only in sequence, but also in the length, So, but still they acquire this centromeric structure. And one constant or conserved mechanism that we have recognized that is responsible for this, organization is the presence of a histone H3 variant in the centromeric region of the chromatin. And that variant, in case of humans or higher eukaryotes, is called CENP-A, which is a variant form of histone H3. So, and this red ovoidals is representing the CENP-A, the histone H3 variant. And that is a constant in every centromeric chromatin region. And that is interspersed with this histone H3 dimethylation at lysine 4, as shown here. Okay? So somehow this region is epigenetically marked to become the centromeric chromatin, in which the histone H3 is replaced by this histone H3 variant called CENP-A. All right? So, obviously, how does it happen? So there is an epigenetic mark. Like I told you, the histone H3 dimethylation and lysine 4, adjacent to which the CENP-A is recruited to replace histone H3. So somehow, following DNA replication, the centromeric region is marked epigenetically, and the centromeric chromatin is assembled with the recruitment of CENP-A. Right? That has to happen. And that has to happen following DNA synthesis. As after the DNA synthesis takes place, this region has to be marked differently and also configured in a different chromatin form with the recruitment of CENP-A. That's not the end of the story. So this is the CENP-A nucleosome. Now next to the CENP-A nucleosome, there are a host of other CEN proteins that are assembled. And again, for operational purposes, this is called, CENP-A NAC. NAC standing for nucleosome associated complex. In other words, so this is the region where the CENP-A nucleosome is organized, and next to it, the CENP-A nucleosome associated complex is organized, and distal to that, there is a CENP-A nucleosome distal protein complex or CAP protein complex organized. Now, all of these proteins have this prefix of CEN, or have this acronym of CEN. You know what CEN for? simply stands for centromeric protein. Okay? So these are a whole host of different centromeric proteins that are organized around the centromeric region. But this all starts only when the centromeric DNA is epigenetically marked with histone H3 modification, like I told you, and then the CENT-A histone H3 variant is recruited. All right? And then that leads to the recruitment of these additional proteins in this manner. So the question is, how is this state of the centromere protected? Well, what is seen is that, the centromeric region typically remains associated with the nucleolus during interphase. And it is thought that this protein called nucleophosphine somehow anchors this centromeric complexes next to the nucleolus during interphase, and as the nuclear envelope breaks down and the nucleolus degrades, this complex is organized in this way, and then the kinetochore is organized next to the centromeric region where the microtubules get embedded. So these are some of the major centromeric proteins that have been investigated for their structure function. CENT-A, that I already told you, is a histone H3 variant, which is essential and conserved in all the centromeric chromatin that have been investigated. Then CENT-B, CENT-C, CENT-G, CENT-H, CENT-U. Again, some of these CENT proteins we still don't know what their functions are, but for some that we know, they are described here. How do we know whether they are essential or not? In two ways. Essentially by knocking them out. So in vitro assays, you can knock them out with short interference RNAs, siRNAs. And I believe, I would assume that most of you know what siRNA technology is, right? You must be knowing. Are you all aware of what siRNA is? Okay. And then of course, many of them have also been identified, for their functional significance, they have been studied by knocking them out in mouse models, right? So when we say that CENT-B is not essential, it means that when you knock it out in vitro, then you still see proper centromeric organization of the chromatin in that region of the chromosome, and chromosome segregation going on without any problem. Similarly, CENT-B knockout mice are also perfectly viable. Okay? So obviously there must be some redundancy in the functional aspects of these proteins. Again, this is another, yes?
Can you say in brief about siRNA technology?
So, essentially, these siRNAs that are designed to match the sequence of the messenger RNAs, and these short interference RNAs, when they, in some regions of the messenger RNAs, if they are able to form a perfect match, then first of all they prevent their translation, and depending on the extent of match, they can also be degraded. The messenger RNA gets degraded. The biochemical details, I can tell you later on, but that's in a very simplistic way the technology that is very commonly used. And but siRNAs are typically transient knockout experiments, but they can also be done in a more stable manner by designing what are called as short hairpin structures, which can be integrated in the chromatin and transcribed and translated into eventually siRNAs that can knock out the messenger RNA.
I think it's not done.
I'm sorry?
I think it's not done. It's not knockout.
Knocked down, yes. And so essentially, so I'm sure you recognize what's the difference between knockout and knockdown, right? So, and you are absolutely right, it's knockdown. Okay, so in another direction, this is how the SEN proteins are organized along the length of the centromeric region of the chromatin, and again in the context of where the kinetochore is organized and where the sphingomicrotubule is attached. Okay, so in addition to that, here's a partial list of some of the kinetochore microtubule binding proteins that again have important functions in microtubule dynamics and also chromatid segregation. So SEN-E is a protein that is in the outer kinetochore, and this interacts with these proteins called BUB-R1, which is a kinase. And in fact, there are reports that SEN-P is responsible for activation of this kinase, and that SEN-P is also a sensor of the microtubule attachment to the kinetochore. And this also senses the occupancy of the tension status, and that is in fact transmitted to the mitotic checkpoint machinery. MCAC is another critical protein which is localized in the centromeric region that typically is involved in depolymerization of the microtubules. Okay? And so what happens is, so long as the proper amphitelic attachment has not occurred and the tension is not sensed from both ends of the cystic kinetochores, MCAC keeps depolymerizing the microtubules from one end, so that the chromatid doesn't move to one pole preferentially. And then, of course, this dynein, dynactine, and so on. There are a few, just some salient functional roles of these proteins listed on the table. So this is how the dependency or interdependency of these SEN proteins was investigated, where you knock down one protein and then you try to see whether or not the other proteins are recruited in the centromeric region. So, for example, if you knock down SEN-P, the positive signs indicate dependency. So when you knock down SEN-P, you see that the SEN-Pi, the Mistral, the Aurora B, they are all failing to recruit to the centromeric region. Okay? Indicating that the SEN-P is in fact somehow responsible for the recruitment of these additional proteins. Even if you cannot see very clearly in the last table, if you look at this table, this shows it in a more clearer fashion, where these arrows indicate again the dependency of these protein recruitments, interdependency of these protein recruitments. So again, SEN-P is at the top, as you can tell, and that is involved in the recruitment of SEN-Pi, SEN-C, and Aurora B. Aurora B is responsible for the function of MCAC. And again, so the arrows pretty much indicates how functionally each of these proteins are interdependent to one another. just a brief mention of this mitotic checkpoint pathway. So, like I mentioned earlier, so long as the kinetochores are unattached, they transduce a diffusible signal that tells that the metaphase-anaphase transition is going to take place. And so these unattached kinetochores are actually the regions from where the signal is generated. How this signal is generated, we still do not know in entirety. But this is again a diagram to suggest some of the major players involved in this mitotic checkpoint function. So here, this chromosome, okay? If you just concentrate on this one, what you see is that the spindle microtubule has attached to only one kinetochore, right? As opposed to this chromosome, where the sister kinetochores are attached to both ends. So here, and so this is indicating that from this unattached kinetochore, a diffusible signal is generated that allows the formation of this complex, all right? And this complex consists of, this is the mitotic checkpoint complex, in which the major players are the BUB-R1, the MAD2, BUB3, and so on. And so long as it is unattached, this complex remains in place. And once this attaches, this signal then tells you that so the SENP is then, it senses the kinetochore, the microtubule attachment, and this transduces the signal to the BUB kinase, and that leads to the dissociation of this entire complex. As a result of this dissociation, the APCC, the E3 ubiquitin ligase that we had mentioned last class, gets activated because the CDC20 protein, which is required for APCC to be active, this E3 ubiquitin ligase to be active, gets released and binds to APCC. As a result of which, now the critical proteins that are responsible for maintenance of metaphase state start getting degraded because of ubiquitination, which are cyclin B1, and similarly on the other hand, securin. Once securin gets degraded, that releases the separase, which is initially in this complex with securin. And then this separase goes and cleaves this protein between the two sister chromatids. And what was this protein? We have talked about cohesin, right? So that cohesin gets degraded, and then the two chromatids separate.
Excuse me. That was my question from earlier. Like I guess, can you go back on the slide, please? Like just between metaphase and anaphase, like say we had non-disjunction, like what exactly is happening? Is it SIMP-E? Is it SIMP-E is connecting to the same side of a centromere, or I'm naming that wrong?
It could be like a lot of things.
Well, that's what I'm trying to figure out. Do we know what it is, or do we not know at all?
There could be a lot of reasons that they don't separate.
Yes, there could be a lot of reasons, but what happens is somehow, if all the proteins are normally synthesized, okay, so there is always a possibility that some protein is not being synthesized, and so the checkpoint itself is dysfunctional. But if the checkpoint is functional, then one common mechanism by which this happens is because of the checkpoint misreading a signal. And how that misreading happens, you know, so essentially what we are talking about is that this chromatid, these two cystic kinetochores has to feel this tension from two ends, all right? Sometimes what happens is, so this is called amphitelic attachment. Sometimes what happens is, if the two poles have not come apart, okay, and the centrosomes are not, so these two poles, you know, these two poles also are organized as the cell is moving from prophase into metaphase. Okay? Sometimes if these poles are actually formed from these structures called centrosomes. Okay? If the centrosome separation is not proper, then and they start forming the microtubule, then what may happen is this may come and join here, but the centrosome is still not at the two ends. Okay? And so the cystic kinetochores may still feel the tension, but the tension may be actually generated out of the same pole. So, but now, like she said, you know, there are many different reasons why segregation defects happen. But essentially, if the checkpoint is not dysfunctional, then somehow the checkpoint gets the wrong signal. That's what it is. And the checkpoint feels that it is ready to push the cells from metaphase, or ready to allow the cells to go from metaphase to anaphase. But somehow that attachment is not proper. You know, if you go and search in PubMed, chromosome segregation, cystic chromatid attachment, you will see that even as we speak, this month, last month, there are papers published. Okay, I saw a pretty decent paper published in this very good journal, PNAS, just within the last couple of months, where a group of investigators have looked at, you know, when does missegregation actually happen? In other words, if a chromosome is lagging behind the rest of the chromosomes, if one chromosome is lagging behind the rest of the chromosomes, does it lead to missegregation? In other words, does it lead to abnormal number of chromosomes in the daughter cells? And this group is saying that perhaps that is not the case. Okay? It has to be something else. The way they did it is because they labeled, you can fluorescently label each chromosome and actually monitor them through time-lapse microscopy. And so what I'm trying to tell you is that, you know, we still do not know all the reasons why segregation defects happen. But we do recognize now the basic mechanisms which are supposed to be controlling chromosome segregation. And any number of, you know, reasons could be there, you know, which might involve, you know, abnormal expression of any of these proteins, or a mislocalization of these proteins, or for that matter, even misreading of the signal generated from these kinetochores that can lead to segregation defects. Yes.
Have you labeled the chromosomes and follow them in real time? Because all the histones and proteins that you are talking about, if the probes are direct to the DNA in an open,
Yes.
what is that?
Yeah, so that's exactly how it is done. So, I mean, we do it also sometimes. Where, for example, you can label one of the histones, say H2B, very commonly labeled, with a GFP, green fluorescent protein. Or you can, you know, you can label them with other fluorochromes.
Okay, but you cannot know if it's 1, 2, 3, 4, 5, they're different. You just count the numbers of dots that you see to know that they are complete. So
you cannot know, I mean, if the chromosome 4 is the one that always
That's right,
replicates.
that's right, that's right, that's right. You cannot, but there are methods in which you can try to label a single chromosome also, okay? But I don't think we are there yet where we can label multiple chromosomes at the same time at our will and follow each chromosome separately. But there are methods, you know, that are being developed with which this is becoming more precise.
So
But-
fluorescent labels, they don't go with the banding patterns?
No, because you know H2B is pretty much distributed all along, right? It's an integral part of the nucleosome. Any questions? No? Yes?
No.
I'm just skipping these slides. This actually talks about a phenomena again, which is an operational term used for you know, defining how these processes are sequentially taking place. In other words, there is this term called licensing, you know, which tells you when does this chromatid, when is the chromatid licensed to undergo segregation. And it is thought that perhaps this licensing happens once the DNA has been replicated. It's almost like you getting a license to drive a car. You know, only when you get that license, you're able to drive. And if you take that license away, you cannot drive. So, in other words, if you prevent certain processes from happening, and it's so in the previous slide, I think what we were trying to show is that this NCM family of proteins play a critical role in deciding when the chromatids are licensed to have the cohesin loaded on them. And so this typically happens during this G1 S transition phase, you know, and only, so during this phase, the cohesin loading is, the licensing takes place for cohesin to be loaded. And during this phase, it cannot load any further cohesin. So, you know, again, experimental evidence suggests that this NCM family of proteins, which is required for DNA replication, has a role to play in the licensing of cohesin loading process. Again, this is shown in the cartoon diagram. Now, just to briefly touch upon the telomere structure. How many of you are aware of the telomere structure? Is there anything specific about telomeres you know about? You know, those who discovered the telomere structural organization, you know, got Nobel Prize just a couple of years ago. Do you know who they are? Have you heard of Elizabeth Blackburn? Anyway, so you know, telomeres have fascinated investigators for decades. You know, for two reasons. One is this protection of the ends of the chromosomes. You know, there has to be some capping mechanism at the ends of the chromosome to protect the telomere. Otherwise, the integrity of the chromosomes cannot be maintained. Second, equally importantly, or perhaps more importantly, a problem called end replication problem. Are you aware of that? Kind of. What is end replication problem? Do you know how DNA replicates? Is there a specific direction in which DNA replicates? Which direction?
3 to 5. 3 to 5. Or
Just
5
the
to
other
3.
way, 5 to 3, yeah, of course. If it is not 3 to 5, 5 to 3, yeah. So it is 5 prime to 3 prime, okay? And if you think about, anyway, before we go into that, let me tell you that, you know, we have recognized that telomeric ends, typically, and this is a conserved sequence, contains a repeat, TTA, GGG. You know, this telomeric repeat sequence is interspersed with other repetitive elements at the ends of the chromosomes, okay? And there is a reason why this repeat is there at the end. And let's just touch upon that. This is end replication problem. So these are the two strands of the DNA, right? So DNA replicates in the 5 prime to 3 prime direction. So no problem for this strand to be replicated, right? The one in black. Because it can go from 5 prime to 3 prime uninterrupted. All right? But what happens to the other one? For this, you need some primer sequences, right? What are also called as Okazaki fragments. Okay, so you have Okazaki fragments here, you synthesize, you have fragments here, synthesize, and then you fill in the gap. All that's fine. But what happens here, at this end? You know, how do you replicate that? So this has been a problem that investigators have grappled with for numbers of years, okay? And now we know how this end replication problem resolved. Because, so anyway, this is just to show you what happens because of the end replication problem. If there is no mechanism to take care of the ends of the chromosomes, you'll start losing the ends of the chromosomes, right? So the telomeres keep getting shorter. And as a result of it, sometimes the synapse, they stop dividing. But in other cases, they go into this critical phase, called crisis phase, in which they either die or they have to reactivate the telomere. And for the telomere to be reactivated, or regenerated rather, there are mechanisms. And in fact, those mechanisms are quite prevalent in cancer cells, which are now replicating in an uncontrolled manner. And the missing piece of the puzzle was identified as an enzyme called telomerase. Okay? And again, let's not go into the details of this one right now. We'll touch upon it in the next class a little more. But telomerase has a catalytic subunit and an RNA subunit. Okay? And that's how telomerases allow the end replication problem to be overcome. This is how it happens. Again, let's not go into all that. There are a whole bunch of other things that have been identified, and we may touch upon them, some of them in the next class, that are all organized or assembled at the telomeric region of the chromosomes. But here is how the telomerase solves the end replication problem. And you can also now recognize why this TTA GGG repeat is present on at the ends of the telomeres that is conserved. So, like I told you, it has a catalytic subunit and an RNA subunit, internal RNA template. This is what happens. So you have this TTA GGG repeats at the ends of the chromosome, right? And this is where the DNA could not be replicated because of, like I told you, you cannot have a primer at the end. Once it is removed, something has to be able to replicate it. So this is how telomerase does it. So the RNA template actually helps solve that problem. So here it is. So this RNA has this sequence, which which is complementary to that TTA-GGG repeat at the ends of the telomeres. Okay? So here it is. So TTA binds to this AAU sequence of the RNA, right? Now, you can synthesize this DNA, right, in the gap. And then it can slide along. So, no, I'm sorry. So, now, once it is bound here, you can synthesize the DNA against this sequence here on the 5' 3' direction, right? And then it can slide along. And then it's, so this one, here the DNA is synthesized, shown in blue, and then this telomerase slides, okay? And you can, again, synthesize the DNA here. Having said that, can you tell me what that catalytic subunit must be? It's now making DNA from an RNA template. What is that enzyme called? Or those classes of enzymes called?
Reverse transcripts.
Right, reverse transcriptase. Okay? So indeed, it's a telomerase contains the reverse transcriptase and the RNA subunit. And that is how the end replication problem is solved. Okay? And so, and once you have synthesized DNA at the end, you know, then you have a primer that can fill in the blanks in between. And so that's how the ends of the DNA gets replicated, the telomere gets replicated. Now it makes sense why you have this TTA-GGG repeat conserved through the evolution as the telomeric repeat, right? And so this obviously you can tell is a monumental discovery. And this is what actually won them the Nobel Prize. Yeah. Yes. How many repeats?
Yes,
Yeah.
how do you know
So
the
this-
idea that you don't
Yeah, so
give
this is interesting. I mean, so actually the mouse telomeric repeats are longer than human telomeric repeats. You know? And I don't know why that is so, and I don't know if anybody knows why that is so. But you can actually resolve telomeric repeat DNA on a gel, and typically, you know, the mouse telomeric repeats are much larger than human. Do you know why? But you know, fascinating work has been done since the discovery of the telomerase. You know, you can knock out telomerase, or you can overexpress telomerase, and you can pretty much immortalize any cell by overexpressing telomerase. You know, typically in a normal cell, the telomerase gets inactivated. Once you overexpress it, telomerase starts, I mean, the cells keep proliferating. So a few years ago, I don't know whether you all were watching TV news or not. No, this was a big news where everybody thought they will stop aging. Okay? And all the networks and all that, you know, had big coverage that now you can stop aging. The flip side is, cancer is nothing but uncontrolled cell division. Too much replication is also a problem. And in fact, there was also a big excitement some years ago about telomerase being a good therapeutic target for cancer. That if you prevent telomerase, you can kill cancer cells because they are the ones which have uncontrolled telomerase activity. Well, it doesn't quite happen that way. I mean, there are problems there too. So I think I have one last slide which says that
But in a normal process, how the cell knows that is, how the enzyme knows that is enough to keep doing the